give a talk Cassava Peel and explored lignocellulose residue for uh, for the for the xyloooligosaccharides production potential treatments. I'm Cristiano Jose de Andrade. I'm food engineering. I earned it in 2008. After that, I ran for the master degree and then to the PhD degree, which I did the split PhD in England, University of Reading. After that, I worked as postdoctoral research fellow in the chemical engineering department, USP. In 2018, I started to work as a professional in the chemical and food engineering department, Federal University of Santa Catarina. And in 2009, I started to work as in the postgraduate program in chemical engineering, which is one of the finest postgraduate programs in Brazil. So basically, we're gonna give a talk about cassava and concept of green chemistry, a brief introduction. After that, we're gonna talk about biosurfactant production and its correlation with cassava and concept of green chemistry. After that, xyloooligosaccharides production, current works, conclusion, and perspectives. So the cassava flour industry is basically the root. This represents the cassava flour processing. So basically the first step is to wash these roots. And of course, this is a residue. After that, you have to cut and peel that. And when you do that, you have a cassava peel as a residue. After that, you can cut and dry this biomass and pressing. When you're pressing that, you wanted to dry this biomass. But when you do that, you release, which is known as cassava wastewater, which is a really rich residue. And after that, a thermal treatment which produce the cassava flour itself. So it's quite a simple process, but it's worth noting that there is at least 3 huge residue which can be explored biotechnologically speaking. It's worth noting that Nigeria, Congo, Thailand, Ghana, and Brazil, they sum up, they together represent more than 56% of the global production. So basically, cassava is related with low-income countries. And as you can see here, when you process cassava root, it generates a lot of residues, solid residues and liquid residues also. So we wrote up proposal, a project to use this cassava peel as a residue and cassava wastewater, and we submit to FAPESC. FAPESC is a provider, financial provider for research in Brazil, in Santa Catarina, and we won about $100,000 to do this research. So basically, the first step is definition of green chemistry. What is green chemistry? According to Environment Protection Agency, United States, green chemistry is the design of chemical products and processes that reduce or eliminate the use of generation of hazardous substance. Green chemistry apply across the life cycle of chemical products, including its design, manufacture, use, and ultimate disposal. So if you think about, if you change a little bit your process, you can make at least greener, not green, but greener, and probably it's worth. So after that, we try to see some trends in the literature, in the scientific trends relating green chemistry with cassava using Scopus database. As you can see, both terms, both subjects are increasing, which is good. That means more information is available right now. But when you look deep in Cassava, most of the data available are related with agriculture, not by biotechnology by itself, but agriculture. So basically, our project is to try to transform or to change a refinery in a biorefinery by using cassava peels to produce xyloooligosaccharides, which is high added value compounds, and also biosurfactant, which is also a high added value compounds. In this case, we are using cassava water as a low-cost culture medium. So there's a plenty of possibilities to apply or to use these residues. I'm going to show you just two of them. So this is cassava water. As you can see, it's colorish, is quite rich in carbohydrates, proteins, and vitamins, minerals, and so on. So it's quite rich. Personally speaking, I've been using that as a culture medium for yeast, fungi, microalgae, and several bacteria. And it's feasible for all of them. So it's quite rich and low-cost culture medium, which is available right now. But before run the fermentation process, the production process, we have to do some pretreatments. The most used pretreatment is to boil that and to centrifuge. When you boil that, you remove some cyanate, which is toxic, but at the same time, you intermix your starch. You make some really big balls of starch. So when you centrify that, you separated the starch from the culture medium that will be used in the fermentation process. So we can run the fermentation process, recover our biosurfactants, purify them using ultrafiltration process. We have doing this kind of work, as you can see. And personally speaking, the most feasible application for biosurfactants is in agriculture as an eco-friendly pesticides. This is just one example of biosurfactants. This is quite well known, surfactant. As you can see, there are 7 amino acids linked, bounded by 10 cells, and 1 fat acids. So if you take a really close look, you'll see L configuration, and D configuration. Personally speaking, this is incredible. It's quite hard to try to construct this kind of structure in the laboratory, I mean, synthetically. So personally speaking, this is a quite remarkable structure. So looks like a really master plan. We have a zero cost culture medium, really low cost culture medium. This is right now is a problem for the industry, and for us it's quite worth, because we can use that. It's rich medium, but our group have been doing this kind of research for over 20 years, and in 2016 we tried to apply the ultrafiltration process in this kind of biosurfactant using cassava water. So in conclusion, there's too much protein. When you think about in purify surfactant or biosurfactants for impurities, you mean to separate them from proteins. In this case, if you think about, there are 2 sources of proteins. The cassava water contain proteins, and the the bacteria, the yeast, will also produce protein. So it's too much protein. So our research group has been working to overcome this situation right now. Our recent publication, this is quite nice journal, we had to do some prospection on membrane-based strategy for downstream processing of surfactant. I mean, we're trying to functionalize this kind of membrane in the sense to improve the separation between biosurfactants and protein. And also, our group have been working in biosurfactant inducers, in particular, two kinds of inducers, hydrophilic and hydrophobic. In addition, we have two chapters book in which we describe the application of biosurfactants as eco-friendly pesticides. And in this one, we have described the potential of cassava wastewater as a low-cost culture medium. I mean, to pigment production, biomass, biogas, any plenty of possibilities to use cassava wastewater as a culture medium. This is some results. We have to try to identify some specific components as a hydrophobic inducers with cassava wastewater, of course, and it changed a lot the microbial growth, of course, but more than the microbial growth and the yield, the production yield, we are interested to correlate this inducer with the structure, the chemical structure, which may change. I mean, the amino acid sequences can be different, can be related with these molecules. So as you can see here is the surface station variation with indirect measurements of biosurfactant production, and they are quite different as well. And we have doing the same approach for hydrophilic inducer. In this case, you have been using some metals, as you can see here, iron, manganese, magnesium, and so on. And on the other hand, we have been doing some progress in ultrafiltration. Personally speaking, this is quite nice, it's quite feasible approach. For instance, the first step is to produce biosurfactants. After that, you have to separate it, microbial biomass from the surfactants. After that, you can run first step ultrafiltration using 100 kilodaltons membrane. This is quite nice because when biosurfactants are at high concentration, they aggregate themselves in a structure like this. They form huge structure like this. When you run some ultrafiltration process, these structures are retentated. On the other hand, salts, small proteins, free amino acids, they permeate it. So basically the first step ultrafiltration retain biosurfactants and also proteins. After that, we can add some solvents as ethanol, methanol, and so on. When you do that, this structure is destabilized. So after that, we can run a second step ultrafiltration. The biosurfactants can permeate it and the proteins can retain it. So it's quite simple, it's feasible. To the best of my knowledge, there is only one company, enterprise in the world doing this kind of process. And most of them have been using polymeric membranes. Our group have been trying to produce some ceramic membranes which can have a wider pH range. They are more resistant to organic solvent, more resistant to chemical cleaning, and so on. There's a clear advantage on the use of ceramic membrane instead polymeric membranes. So this is just one residue. The second one is cassava peel, which can be used as a source of xyloooligosaccharides. basically this is lignocellulosic materials. For instance, sugarcane bagasse, corn cob, wet straw, and also cassava peel. So basically the idea is to get the end cellulose, to cut that, and to produce SHOS. SHOS is basically a prebiotic compound. That means it favors bifidum bacteria and beneficial bacteria for our health. And it can be produced from cassava peel. So this is a really picture. It's a picture of cassava processing, flour processing, which has been providing this cassava peel for us. Our group have been trying to facing, it's quite challenging actually, to produce SHOS from cassava peel because there is too much starch. So basically our group has been trying to remove this starch from cassava peel. We have been trying to do some low-cost approach and have been doing some progress, I would say, but we are not that close to produce xyloooligosaccharides from cassava peel right now. This is my research team. We have a postdoctor, master students, PhD students, teacher, professor, and so on. So they do the all job, and I'm really thankful for them for that. We have to conclude that to produce xyloooligosaccharides from cassava peel, probably in this year, right now. So cross fingers. And would you like to apply the concept of green chemistry to food industry? This is my email contact. Lattice ResearchGate, which is a really nice one, or Seed. And thanks so much for your attention.